Alright class, this is the beginning of the section of our course focused on calculus. Hopefully most of you have taken some calculus before. If you have, this toolbox should help to reinforce your calculus knowledge. If you haven't had calculus before, this and the following toolbox will give you an entry into the basics of calculus, with the real focus on why we want to know calculus and how we use calculus. Calculus can seem really dry when taught by mathematicians, but really calculus is the study of things that change. And of course, things are changing rapidly in our environment today, so we need the mathematical tools necessary to study those changes. I would also like to take 1 minute to note something important. A lot of math courses spend a lot of time on complex theorems and derivations for computing the derivatives of different elementary functions, such as the trigonometric or the logarithmic functions. That's not what the next few toolboxes are going to be about because mathematicians have already done that, so you don't have to. In fact, there are derivative and integral calculators online that you can use to compute derivatives and integrals for yourself, and I use them routinely to check my math. So we're not going to focus on the derivations. We're going to focus on using derivatives and understanding them as mathematical concepts that help us study a changing environment. The definition of a derivative is that the derivative of f of x is equal to the limit as h approaches 0 of f of x plus h minus f of x, all divided by h. That may sound like fancy math speak, but if you take some time to work through some real-world examples, it's really not. And so we're going to do that in a minute. But first I wanted to suggest some alternate ways of defining the derivative that may make more intuitive sense to you. For instance, we could also state that the derivative is the relative rates at which two different things change. Or in other words, it is how fast one thing changes with respect to how fast something else changes. Or we could define a derivative in the way that many biologists think of it by saying that the derivative with respect to time of the population of any organism is simply its net growth rate. Relating back to the derivative above, this is the relative rate at which the population increases or decreases in relation to the relative rate at which time passes. Or we could use a definition that's suited to race car drivers. The derivative with respect to time of the position of a race car is simply its velocity. Different calculus textbooks use different notation for writing out the derivative. Some will write that the derivative of f of x is f prime of x. Others will write that the derivative of x is df dx. I will always use the second notation, df dx, because it is more generalizable, as we will see throughout our calculus toolboxes. However, I want to make sure that you're aware that you might see the derivative written as f prime of x elsewhere. Okay, now let's actually explore that first definition using the kind of problem that we might actually encounter in our research. One of my friends, Sven Krantz, studies a microscopic algae called Crania Brevis, which is shown in the picture on the top right of this slide. Crania Brevis is a protist with really interesting ecological and behavioral patterns. But that's not the reason most people care about Crania. They care about Crania because it is the algal species that causes most of the harmful red tides along Florida's coast. It produces a toxin called brevetoxin that kills fish, leading to major fish die-offs along our coasts. When the red tides are too bad, the brevetoxin can also become aerosolized, making it harmful for us to even walk near the beach. The brevetoxin also bioaccumulates in herbivores and predators in the marine ecosystem, and as it accumulates in shellfish like clams and oysters, those fisheries have to be shut down as a result of all the brevetoxin that makes the shellfish poisonous to humans. So these algae can be quite an environmental problem for humans, and hence Sven is studying their biology to try to predict where and when the blooms will form and how long they will last. Sven grows these algae in his lab, and one of the most basic things he's interested in knowing is what their growth rate is. So let's talk about the growth rate. To measure the growth rate of Crania, Sven tells his grad students to grow them in the lab. The grad student puts them in an Erlenmeyer flask with nutrient-enriched seawater and then watches them grow over time and measures their abundance while they grow. Now, let's say the grad student was feeling fairly lazy and hence decided to measure the growth rate only once per week. Well, the grad student might wind up with a plot that looks like this. This plot shows us the abundance of cells per milliliter of seawater on the y-axis as a function of time on the x-axis. So we can call this f of t. F of t is therefore the abundance of the algae as a function of time. If we want to compute the net growth rate, we simply have to ask the question, how fast did the population grow? More precisely, we say that the net growth rate is equal to the change in abundance divided by the change in time, which is also equal to the change in our function f of t divided by the change in t. Note that here I'm using the common notation that the Greek letter capital delta means change in something. So that is the change in f of t divided by the change in time. Alright, let's calculate that for the first week. At the start of the experiment, there were 40,000 cells per milliliter, and after 1 week the grad student measured 127,873 cells per milliliter. So to calculate the growth rate, we take 127,873 cells minus 40,000 cells divided by the 7 days that have passed. Graphically, we can think of this as the amount that the population has gone up divided by the number of days that have passed, or in other words, the slope between those 2 points. We can do this again to calculate the net growth rate during the second week of the experiment. After week 2, we measured 401,529 cells per milliliter, so the population increased from 127,873 cells per milliliter to 401,529 cells per milliliter over 7 days, and we compute a growth rate of 39,094 cells per milliliter. For week 3, we do the same thing and calculate a net growth rate of 109,300 cells per milliliter per day. For week 4, we get 121,978 cells per milliliter, and for week 5, we get 2,787 cells per milliliter per day. Okay, looking at that, we see that the growth rate changed a lot over the course of our experiment. As biologists, we want to know why those net growth rates changed so much, because ultimately those changing growth rates are what determine whether this harmful algae takes over and forms a huge bloom affecting beachgoers, fish, and fishers, or whether it just peters out harmlessly. So let's graph those growth rates really quickly. Okay, this graph shows a bar plot of the net growth rate as a function of time for those 5 weeks. Another way of putting it is that those net growth rates are nothing more than the slopes of those 5 red lines in the figure on the left. That net growth rate really varies though. It starts off with a relatively low growth rate, then it starts to grow pretty quickly, before it eventually drops off to a very low growth rate. But figuring out what is driving this can be pretty difficult, in part because we've only measured the population abundance, and hence the growth rate, once per week, because our grad student was kind of lazy. What if our grad student went out and instead made the measurements once every 3 days? Okay, now we've got a graph with higher temporal resolution. We've got measurements of the abundance of Crania every 3 days, and so we can go through the same process. We can calculate the net growth rate, which is the increase in abundance between 2 measurements divided by the 3 days that have passed, or in other words, the slope of the lines between 2 points. Because we've made our measurements more frequently, we now have a total of 11 growth rate measurements over 5 weeks, instead of just 5 measurements. Now let's graph these growth rates. Alright, we're starting to get a better picture of a slow ramping up of growth rates, followed by a rapid decline in growth rate. But let's make that grad student do a bit more work. Let's make the grad student go in every day and measure the population abundance. It really isn't that hard. They just take a 1 milliliter sample and they put it into an instrument called a flow cytometer. Whole process should only take about half an hour, so it's reasonable to ask them to make the measurement every day. Well, let's see what we get if they do. Alright, this is starting to look a lot better. We're getting a pretty smooth abundance curve. And we're also starting to get a much smoother net growth rate curve as well. Still a little bit steep on the right side though. That last week sees a really abrupt drop off in growth rate, so maybe we want the grad student to measure this even more frequently. But first, let's think about how our growth rate measurements relate to that definition of the derivative above. Okay, these were our 3 growth rate plots that we've generated so far. Remember that we're calling our population as a function of time the function f of t. So what did we do to calculate these plots? Well, to calculate the first growth rate plot, we computed the difference in the population between our measurements made 1 week apart, divided by the 7 days that separated those measurements. In other words, we calculated the abundance at time t plus 7 minus the abundance at time t, all divided by 7. Or in our math speak, we computed f of t plus 7 minus f of t divided by 7. In the second plot, with our measurements made every 3 days, what we really computed is the abundance at time t plus 3 minus the abundance at time t, all divided by 3. Or in other words, f of t plus 3 minus f of t divided by 3. And in our third growth rate plot, we made measurements every day and computed the growth rate as f of t plus 1 minus f of t divided by 1. So let's compare that to the definition we have above. Our definition tells us that the derivative is the limit as h goes to 0 of f of x plus h minus f of x divided by h. Let's just substitute t in instead of x for a moment, since we've been looking at something that is a function of time and calling it f of t. Well, if we ignore that little thing that says the limit as h approaches 0, then we can see this equation looks very much like our growth rate equations. The plot on the left is simply that equation if we substituted in 7 instead of h. The plot in the middle is what we would get if we substituted in 3 instead of h. And the plot on the right is what we would get if we substituted in 1 instead of h. So looking at this, we can see that our equation for net growth rate is pretty similar to the equation for the derivative. We can also see something really important. As we make h smaller, or in other words, as we make our measurements more frequently, we get more accurate results. Let me say that again. As h gets smaller, we start to get more accurate results of how the growth rate is changing in time. If we get more accurate results by making h smaller, why don't we tell our grad student to go make the measurements every 6 hours instead of just every day? Alright, now h is 1/4 of a day, or in other words, 0.25. And we've got a lot of measurements, and this is starting to look more accurate. We've got a nice growth curve here. But what if we wanted to get much, much better using technology? If we set things up right, we can actually connect our flow cytometer right up to our Erlenmeyer's flask with our Crania growing, and we can get a continuous measurement. Basically, we are making the time between measurements almost infinitesimally small, maybe even less than a second. What we've actually done is taken h and made it get really, really, really small, less than a second. We've made the time between measurements approach 0. Or in other words, we've calculated the derivative of the abundance with respect to time. We've calculated the derivative of f of t with respect to time. We've basically calculated the growth rate over instantaneously short periods. This is really what the derivative is. It's simply figuring out the rate at which one thing changes with respect to changes in another. In this case, we're asking how fast does the population change with respect to how fast time is passing. And we're calculating this instantaneously to get really accurate results. The next thing that I want to talk about is the geometric definition of the derivative. Remember from before that when we were calculating the growth rate, we were basically just taking the slope between 2 points on our graph of abundance versus time. And the growth rate that we calculated was really equivalent to the derivative if we calculate the growth rate over a very small time interval. That brings us to the geometric definition of the derivative. The geometric definition of the derivative tells us that the derivative is equal to the slope of a line drawn through a point on the curve that is tangent to the line. So basically, if we pick any point on our function, which I'm illustrating here with that moving yellow point, we basically draw a line that nestles up against our function, going through that yellow point, and with a slope that is equal to the slope of the function right at that point. So if we watch this animation play through, we see that as that red line becomes steep, the population is increasing rapidly, and our net growth rate, which equals our derivative, is higher on the graph on the right. On the other hand, when the slope becomes much shallower, the net growth rate starts to drop. This is what is meant by the geometric definition of a derivative. The derivative at any point is equal to the slope of the tangent line going through that point, which is shown in red in this graph. The geometric definition of a derivative is not a definition that we typically use to compute a derivative, but it's a great definition to keep in mind if you want to test for yourself to see if the derivative that you've calculated actually makes sense. Okay, so far we've learned how the derivative is related to the growth rate of the phytoplankton. But how can we actually use the derivative to learn something useful? Well, one thing that we can do is we can take that derivative of f with respect to t that we calculated, and instead of plotting it against time like we did before, let's plot df dt as a function of the abundance of Crania in our experiment. This now tells us how the net growth rate varies as a function of the population size of the Crania, and this gives us some useful information. We see that the net growth rate grows almost linearly with abundance at the beginning of the experiment, and then plateaus and declines rapidly. But let's actually try something a bit different. Let's calculate the specific growth rate. The specific growth rate is the number of divisions per cell per day, or in other words, it is a measure of how rapidly the population is doubling. The specific growth rate equals the net growth rate divided by the population size. So basically, it is normalizing the growth rate to the size of the population. Another way of writing it would be to say that the net growth rate is equal to the derivative of f with respect to t divided by the function f. If we go ahead and plot the specific growth rate as a function of time, we see that the specific growth rate, or in other words, how fast each individual Crania cell is growing and replicating itself, declines throughout the experiment. It starts off relatively high and declines slowly for the first couple of weeks, but during the fourth week the specific growth rate declines really quickly until the specific growth rate is nearly zero by day 30. You should of course be asking yourself, why? What is causing this pattern, and what does it tell us about this harmful algal bloom in the real ocean? Well, instead of just plotting specific growth rate against time, let's try plotting specific growth rate against the nutrient concentration in the incubation flask, which hopefully the grad student was measuring at the same time. Okay, now we're really getting somewhere. This plot shows us the specific growth rate, or in other words, df dt divided by f, as a function of the nutrient concentration, and it gives us some useful information. The growth rates are low when nutrient concentrations are low, and they are high when nutrient concentrations are high. We can also see a functional relationship between specific growth rate and nutrient concentration. It shows a saturating relationship that we often refer to as a monad relationship. This saturating relationship suggests that at very low nutrient concentrations, adding a little bit of nutrients leads to a rapid increase in growth. But at higher nutrient concentrations, the effect tends to saturate, and we reach a point at which adding more nutrients no longer makes much of a difference in the growth rate. Alright, let's go through another example of finding the derivative. This time, let's focus on the toxin produced by this little tiny algae, since after all, it's that toxin that causes all the problems for humans. Brevetoxin is a neurotoxin that binds to the sodium channels in animal nerve cells. This leads to a disruption of normal neurological processes and causes an illness known as neurotoxic shellfish poisoning. The structure of this molecule is shown in this slide. So basically, that molecule there produced by the little algae up at the top right, which is only 20 micron in size, causes all of those problems that shut down our beaches, poison our shellfish fisheries, and kill tons of fish and even sea turtles. So let's tell the grad student to run the same Crania growth experiment, but this time let's tell her to measure brevetoxin concentrations as well. If the measurements are made once per week, like they were at the beginning last time, we can plot the concentration of brevetoxin as a function of time and call this function g of t. Then if we compute the slopes of the lines connecting any of those points, we find the rate of change of brevetoxin with time. But of course that's a very crude estimate of how the growth rate is changing in time, so we might do the same experiment while making measurements every 3 days. And then we get this plot. Or we could figure out a way to make the measurements continuously and get this plot, which now gives us the true derivative, since we are looking at instantaneous rates of change. Or in other words, we're calculating the rate of change over an infinitesimally small time period. This shows us how brevetoxin concentrations and the rate of change of brevetoxin vary in time. But these aren't the only things that we could compute the derivative of. What if, instead of plotting brevetoxin just against time, we took advantage of the fact that we also measured nutrient concentration at the same time and plotted brevetoxin as a function of nutrient concentration in our experiment? Our plot of brevetoxin concentration as a function of nutrient concentration basically shows an inverse correlation. At the beginning of the experiment, we were actually in the bottom right corner of this plot with high nutrients and low concentrations of brevetoxin. Over the course of the incubation, however, the nutrients were used up and the brevetoxin increased, so that by the end of the experiment, we had low nitrate and high brevetoxin. To compute the derivative of brevetoxin concentration with respect to nitrate concentration, or in other words, the derivative dg dn, all we need to do is figure out how the slope of that line changes. And as you can see, it is basically linear for most of the graph. So the derivative is basically constant at -2 until you get to very high nutrient concentrations at the very beginning of the experiment. You'll note that this derivative is always negative because brevetoxin goes down when nitrate goes up, and vice versa. You can think of this derivative as telling you how brevetoxin varied with respect to variations in nitrate, or in other words, it tells you how much brevetoxin the Crania used up for each micromole of nitrate that they utilized as a nutrient. Since the derivative dg dn is basically -2 most of the time, that tells us that the Crania in our culture were usually producing 2 nanograms of brevetoxin for every micromole of nitrate that they took up. That is a potentially useful piece of information for studying the impact of brevetoxin in coastal ecosystems. Alright, next let's talk about the second derivative. And let's talk about it while thinking about launching some heavy rocks with a catapult. But first, let's define the second derivative. The second derivative of a function is simply the derivative of that function. So to calculate the second derivative, you just calculate the first derivative, and then you calculate the derivative of that derivative. If we were talking about something like a race car, the first derivative of the position or location of that race car is the race car's velocity, and the second derivative is the acceleration of the race car, which equates to the rate of change of the velocity. On the other hand, if we think about the second derivative graphically, the second derivative corresponds to the curvature or concavity of a function. If the curve is concave up, that means its second derivative is positive. If the curve is concave down, that means the second derivative is negative. Let's get back to that catapult. If we launch a boulder with a catapult, we know that it will follow a parabolic trajectory. It'll head up and away, reach the peak of the parabola, and then start to descend and hopefully land on the heads of the attackers besieging our city. But why rely on hope when we can use math to figure out where it will land? Now of course we've got this plot here showing height as a function of distance. Let's call the function that describes this plot h of x for height as a function of x. We can compute the derivative of this function from the slope of the graph. The graph on the left shows the tangent line of our function, while the graph on the right shows the slope of that line, which is equal to the derivative dh dx. This derivative is quantifying how rapidly the height of the boulder changes with respect to distance as we move along the path of our parabola. We can see that the derivative decreases constantly along the path. This should make some sense based on how the slope of that tangent line was changing, and this can help us calculate our trajectories to hit that enemy army. But I'm a scientist. I don't care about hitting enemy armies. I care about learning how the Earth system actually works. And I can take and use this same data to learn something about the Earth. But first I'm going to plot height against not distance, but time. Now we have a plot of height versus time on the left, and we've got a plot of the derivative of height versus time on the right. The derivative of height versus time is nothing more than the downward component of the velocity vector. So it tells us how fast our boulder is traveling either up or down. When we first launched it, at time t equals 0, it was traveling up at 10 meters per second. However, its velocity rapidly slows down, and when it hits the ground, it's traveling at negative 10 meters per second. We can see that the derivative, or the velocity, is decreasing as a linear function of time. Next, let's plot the second derivative. Remember that the second derivative is simply the derivative of the function. So really, we're looking for the derivative of that function we've graphed on the right. So we'll move that graph over to the left and compute its derivative. Since dh dt is simply a line with a slope of negative 9.8, we can see that its derivative, which is the second derivative of h of t, is simply the constant value negative 9.8. This should be familiar to everyone who has taken physics. This is simply the acceleration due to gravity at the Earth's surface. Calculating the second derivative of our boulder's height with respect to time has allowed us to compute this important value. One really important part of understanding how a derivative works is the ability to estimate what the derivative of a function should be. This is a really useful skill because it allows us to check our math when we're not quite sure if we're right. In order to build our intuition for the derivative, let's practice with a few of the functions that we've already dealt with in other circumstances. First, let's take a look at the sine function, which we can use to study the height of waves as they pass by us on a dock. If we look at the sine function, which I've graphed in the top plot, you can see that the slope of the sine function is initially positive. At the point x equals 0, sine of x is increasing, so we know the derivative should be positive. And this shows up in the derivative, which starts out positive. The derivative starts to decline, however, and by the time we reach pi over 2 and find that sine has hit its maximum value of 1, the derivative must reach a value of 0 since the sine function is no longer increasing. As sine of x starts to decline then, the derivative becomes negative. As sine of x crosses 0 again, its slope becomes as negative as it will ever be, so the derivative reaches its minimum of negative 1. After that, the derivative starts to come back up as the slope becomes less steep. At a value of x equals 3 pi over 2, sine of x bottoms out at negative 1 and starts to increase, so the derivative must be positive again. This process repeats itself, following the repeating pattern of sine. The derivative thus always stays between negative 1 and 1 and has the same periodicity as sine, although it is out of phase, meaning that it's shifted slightly to the left. In fact, the derivative of the sine function is the cosine function, which you might have been able to guess just from looking at this graph. Next, let's talk about the exponential function, which we've encountered when studying population growth rates and looking at how rabbits grow when they're breeding like rabbits. The exponential function starts off by increasing slowly, but it continues to increase faster and faster as it goes along. This means that the slope gets steeper and steeper, and the derivative also becomes larger and larger over time. This makes sense with what we know about exponential growth. The more rabbits there are, the higher the growth rate will be, so the higher the derivative is. Both the rabbit population size and the derivative continue to increase in time. In fact, it turns out that the derivative of e to the x is actually e to the x. So both of those graphs look exactly identical. This is actually the reason that we have that special value of e equals 2.7183. E is a special value that is the only value for which a function equaling a constant raised to the x power will have a derivative equal to that original function. Finally, let's look at a polynomial function. I've chosen the relatively simple polynomial 0.5x cubed minus 3x squared plus 2x minus 5. This function starts off negative but begins to rise a little bit, so we know the derivative has to start off positive. However, after a value of about x equals 0.5, we can see that it starts to decline and the derivative must be negative after 0.5. The derivative will stay negative as long as the slope of f of x is negative. And looking at that graph on top, we should expect the derivative to stay negative until somewhere between x equals 3 and 4, when the slope flattens out and then becomes positive. As we continue further to the right, we see that f of x is concave up, so the derivative continues to increase with increasing x after 4. Okay, hopefully this toolbox has helped you build up some intuition about derivatives. Remember, the derivative is nothing more than the rate of change of one thing with respect to change in something else. Most of the time we think about a derivative with respect to time, in which case we're simply asking the question, how fast is something changing in time? And geometrically, we can figure out what the derivative should be, because the derivative is simply the slope of a tangent line drawn parallel to the curve, like we've seen in these animations. Meanwhile, the second derivative is simply found by taking the derivative of the derivative. Okay, that's it for this toolbox. In the next toolbox, we'll go through the math that's necessary for calculating these derivatives. But first, make sure to take the online quiz.